Dear students, in the previous lecture on environmental genomics, I taught you about 4 different generations of sequencing techniques. The first generation sequencing Sanger technique developed by Fred Sanger, Nobel laureate. The second generation sequencing techniques where we multiplexed and thus reduced the net cost of sequencing a base pair. The third generation sequencing technique where we moved from optical measurements to electrochemical measurements. The ion torrent, for example, uses what is called as the world's smallest pH probe, pH meter, to sequence to get the signal when we are sequencing the genetic element. And then the fourth generation sequencing technique of nanopore sequencing, where the DNA is sequenced as it passes through a nanopore, very small pore in a protein in a membrane. And then I went ahead and told you about ORFs, how ORFs are identified by computers, and then how computers proceed forward to analyze the genes, to analyze what is present. Now, in order to understand how computers move ahead after they have identified the ORFs and after they have assembled, we briefly talked about assembling. The next step usually is alignment. So, in environmental genomics, we don't need to fill in the gaps as we need to do if we were working with pure microbiological questions or pure biotechnological questions. We just need to understand what genes are present and what sequence they are present. So, we will get an idea of what sequence they are translated in, and if they are triggered at the same time, if they are upregulated, downregulated together, and what these fancy upregulation, downregulation terms are. So, for applied environmental microbiological questions, we don't need to fill in the gaps in the genome. If we get the genetic information, the sequences, we want to assemble them so they are more meaningful. So, we get multiple genes in a single scaffold. So, that is the assembly part which I was talking about last time. So, let us start from there. So, in assembly we use different processes, different algorithms. Some of them look at the similarity in different sequences, look at the overlaps and how frequently the overlaps happen. If there is one overlap that happens only one time, it might probably just be a coincidence, but if there is another overlap that happens thousands of times in a pool of genetic sequences, we might assume that okay, they were together and then they were split from different places and then they were amplified and sequenced. So, probably we can attach these two together, we can attach the two genetic sequences together. And we talked about it. The other option when it comes to assembly is de novo sequencing. The other option in sequencing is we look at algorithms that will predict what the next nucleotide should be. So, there are some really good algorithms that can actually predict the next nucleotide, and we ask them to predict, okay, what should follow G, what should follow, okay, let us say A should follow G, what should follow A, okay, C, T. So, when they predict this, we look for where in this pool of genetic sequences are we seeing same sequence that it is predicting, and if it is if we find it and we will notice that there is also some overlap, there is some statistical significance, and it makes sense to combine, assemble them together, then they are assembled together into a long sequence. So, remember most sequencing techniques will give you anywhere from 60 base pairs to 600 base pairs, maybe in case of Sanger up to 2000 base pair, but then you need to assemble them and make really long scaffolds. So, once the assembly has been done, the next step for an environmental microbiologist is to align it with the given databases. So, what people have done very painstakingly over last few decades is that they grew microbes in the lab, they cultured them and they understood their behavior, they sequenced them and they made libraries of their sequence. And what they found out was they found each ORF in those complete sequences, they expressed the protein specific to the ORFs and they found out the nature of the protein. So, they know, okay, this particular ORF encodes for protein that helps in lactose metabolism, this helps in that helps in, so they found this makes efflux pumps which get rid of heavy metals in the cell, the heavy metals in the environment that find a way inside, it effluxes them out. So, for each ORF, for each protein, they found out the sense. So, a lot of it is already known for microbes that have been well cultured in lab for many years now. So, all this information is collectively and very nicely arranged together in databases, and some of them are very well curated databases. So, we are very assured of their quality. Also, nowadays it is required that if you sequence something, you need to submit your sequence to GenBank. So, I will write these name down here because I will write these names here because they are very important. So, you have GenBank. And as the name suggests, GenBank is a bank of all genetic sequences. So if you sequence something to publish your work or to communicate it with public and with other scientists, you are required to submit your sequences in GenBank, and they have a very streamlined process for different kind of sequences. And one of the homeworks would actually require going through the GenBank, looking at their submission processes for different kinds of sequences, trying to understand them and answering some of the questions. So this is GenBank for you. So after you have, now what people can do is, when you submit your data in GenBank, you also submit metadata. For example, you will inform what kind of sample is it. Is it an environmental lake sample, or is it something you grew in the culture? It's something you isolated from a petroleum reservoir, or it's something that was growing on in your bathroom tile, right? So you give some information. So if I sequence something and I assemble a very long sequence, and let's say it matches really well, so this is called alignment. So I do alignment. What alignment means is that base pair by base pair. So base pair by base pair, the sequences are aligned, the one that I am submitting or the one I have recently sequenced with the one that's already present in some database or GenBank or other similar bank of genetic sequences, and then they calculate percent similarities. And let's say, so then we can calculate percent similarity. Let's say it is very similar to something that has been submitted to GenBank and was found in a groundwater aquifer which had perchlorate contamination. So then I know that this sequence was found in groundwater aquifer that had perchlorate sequence, and maybe there is then I'll get some information about what I can expect about the sequence that I just sequenced. Let's say I got this sequence from from a groundwater aquifer, but I don't know if there's perchlorate contamination or not. But I noticed that every time people submitted a similar sequence to GenBank, it was from perchlorate contaminated soils and aquifers. Then I can say there's a very high probability, or there is at least some good probability, that this well from where I got this aquifer from where I got my sequence also has perchlorate contamination. So the GenBank serves a very nice purpose. It gives us a very bird's eye view idea of where else such sequences have been found. However, GenBank is not very well curated. Its purpose is not alignment like this. Its purpose is to just collect all the sequences, to maintain transparency in research, and to prepare a repository of Earth microbiome. So Earth microbiome, very briefly, is a project where we are trying to sequence everything that's alive on Earth, every sequence that's available on Earth, whether it is alive bacteria, eukaryote, archaea, higher order of life like ourselves. We have been fully sequenced, by the way, or whether it is the sub-alive, sub-dead viral particles. So everything that can be sequenced, they want to sequence, and they want to make a bank out of it. So we know what the genetic code of life on Earth is. That is the whole purpose of GenBank. Okay, so this alignment, we do it by databases that are whose purpose is to align and to give information. One of the most common one is BLAST. Another homework from in this week is based on BLAST, where I'll give you the FASTA file. First you have to combine the FASTA file, and then you have to BLAST it, which means you have to align it. And when you align it, it will give you an idea of the percent similarities with different submissions. What the submissions are, in many cases people are submitting from pure cultures, and they know, okay, this is Clostridium thermocellum. So if it has a very good match with Clostridium thermocellum, you know it's probably Clostridium thermocellum or something very similar. So this way you can give an identity to your sequence, or you can get some idea about your sequence. Let's say your sequence, okay, now in blasting we can do in two ways. We can do BLAST X, we can do BLAST P, and you can do BLAST N. So let's look at BLAST N and BLAST P, because they are two most widely used commands in BLAST. So I want to take a segue back to the BLAST in general. You can align your sequences using BLAST repository, either online on their web page if you have limited number of sequences. But if you have created lot of sequences by the wonderful new generation, next generation sequencing techniques, then the recommendation is you download the database, the BLAST NCBI data. By the way, yeah, the BLAST database is maintained by NCBI. So you can download the NCBI BLAST database and then do the alignment locally on your server, on your computer. So BLAST N and BLAST P. BLAST N, you look for alignment in the nucleotide. So A for A, T for T, G for G, C for C. In BLAST P, you do a protein query. So you take your ORF, you can translate it into a protein in silico obviously, and then you look for protein matches. This is BLAST P is very helpful when we have degeneracy. So multiple codons coding for a single amino acid, we know that codon bias. In degeneracy, what happens is that it doesn't matter if a codon changes here or there or not, but the amino acid would be same. So the BLAST N will tell you, well, the similarity in the nucleotide is not very much, but if you do BLAST P, you can notice that, oh, the protein similarity is very high. So that is BLAST P and BLAST N for you. And now let's move on to what we notice in our real life samples when we are trying to do alignment and annotation. Oh, I should mention this, that alignment is looking for similarities, which we do with BLAST. Annotation is when we try to give it a taxonomic identity. So we have some really well curated databases, and like green genes, for example, we can use RDP, ribosomal database project, we can use green genes, and Silva, we can use these databases for annotating our sequences after we have aligned. So sometimes in NCBI BLAST we align, but we cannot annotate because many of the sequences entries in NCBI BLAST are unknown bacterium from Gulf of Mexico sand, unknown bacterium from Arctic. So we cannot annotate, we cannot give it a name. But green genes, for example, Silva, for example, RDP, they maintain a very well curated repository of known, classified and well annotated microbes. Now when we sequence and we create a large amount of sequence data, we run into an issue that are we sequencing deep enough? Which is, are we capturing the entire diversity, are we capturing all kind of ORFs that are present in the sample or not? The other possibility is that I capture a lot of ORFs, but the ORFs, I don't have the information about them. So I captured the ORF, but I don't know what kind of protein it encodes for. It is also possible that it is a nonsensical ORF. So let's look here. One of the things about number of ORFs that we will capture from a sequencing is what is the size of the genome that we are sequencing. For example, if we have an organism that is 3 megabase pair, genome has 3 megabase pairs, then we can predict less than 3000 total ORFs. But if we are sequencing something that has more than 8 megabase pair size of genome, then we should expect up to 7000 ORF. So as the genome size increases, number of ORF increases. However, after some time we notice that's not the case. For example, here there are very few members that have very high base pair and have very high number of ORF. Now in this, the other problem is uncharacterized ORF. The alignment and annotation work on the principle that if the genetic sequence is the same, or if the protein amino acid sequence is the same in case of BLAST P, then the function will also be same. It works on this assumption. So if, let's say, I characterized a genome from a bacterial genome, and I characterized an ORF, and I decided this ORF is very important for SOS system, save our soul signal. So this is when the bacteria undergoes a panic mode and it turns on certain genes to increase its survival rate. Now let's say in a new mammalian species the same ORF is found, so we can assume that it has same function in a eukaryotic system, just because their nucleotide sequence or their amino acid sequence is highly similar. Okay. But many a times we, as I mentioned earlier, we don't have the information about the ORFs that we are noticing. Especially this is a trouble now because we are generating such high amount of genetic sequence, and we are characterizing so many ORFs. We do not know, we don't have complete information about many of them. The other problem is we find an ORF, but it's a non-coding RNA. A very classic example would be 16S rRNA gene. Now 16S rRNA gene is a ribosomal nucleic acid, obviously, but ribosomal RNA, but it's a non-coding in sense that it does not code for a protein. That's also possible. The other thing we have to understand is that let's say I'm doing whole genome sequencing of a particular microbe, and I'm interested in genes that trigger lactose degradation, but most of the genes in the bacteria are not related to lactose degradation. Most of them will be housekeeping genes that are used for day-to-day business, or that are used for making efflux pumps, or that are used for replication. So my genes of interest are very small proportion usually of the entire genome. Thus, another question arises: do we really need to do whole genome sequencing, or can we just do amplicon sequencing and figure what we need to figure out? So on the left here we have a panel that is showing as the number of ORFs increases, how number of ORFs related to DNA replication, translation, transcription, signal transduction and energy generation changes. So for smaller ORF per genome, we have most of the ORFs dedicated for translation, few for transcription, and for DNA replication. Those cells says my priority is that I should be able to replicate, and whatever mRNA I make, I should be able to translate it very quickly. So one single copy of mRNA should make enough proteins. I don't need to make multiple mRNA. So ORFs for transcription are smaller. Same is for signal transduction, and energy generation remains constant, because all of them, no matter how big the genome is, how many ORFs they have, they require, everybody knows energy is important. Now here we have a very nice, very beautiful pathway, pentose phosphate pathway, showing how glucose undergoes different degradation steps to give energy, to create energy carriers in the cell. Now this is a very complex system, but this is a very simple schematic of even more complex biochemical reaction chain. And this is to give you an idea that at each step there are multiple enzymes involved. Each of these enzymes are encoded by certain ORFs, and thus just for this pentose phosphate pathway, we have multiple ORFs that we'll have. So in fact, one of the limitations we talk about when we talk about metagenomics or whole sample genomics in environmental engineering is that our functional genes of interest are very, very small proportion of the entire genome. Most of it is this housekeeping genes. Now the other thing is, we are talking about the size of the gene. So the size of gene varies, size of genome varies. So in the inside here we have different kinds of bacteria, gamma proteobacteria, molecules, beta proteobacteria, Bacteroidetes and alpha proteobacteria. So look here, even among the proteobacteria we notice that the size of genome changes. These two are beta proteobacteria, but the size changes. And then the big one is the mycoplasma, and look how big the genome size is. So when we have a larger genome size, we'll have more ORFs, and many of them would probably be uncharacterized. If we are lucky enough, they are characterized already. If not, we'll have to do it ourselves. And the other thing is that we'll have very small proportion of functional genes. Now when we're talking about functional genes, let's move to functional genomics. As environmental engineers, environmental scientists, environmental students, or students of environmental science, we are very interested in functional genomics, because it really doesn't matter for us a lot if all the pentose phosphate, how many copies of pentose phosphate system are present, how many ORFs related to pentose phosphate system are present in the microbe. But it's very important for us to know, for example, is this microbe resistant to heavy metals? Is this microbe resistant to mercury, methyl mercury, or is it resistant to some antimicrobials, antibiotics? So that kind of information is more important. So we go into functional genomics. We are more interested in the function. Now before I go into functional genomics, let's look at the omics terminology. So whether it is metagenomics or genomics, we are talking about omics. All these have suffixes as omics. So let's look at them one by one. When we are sequencing DNA, we have 4 different kinds of omics: genomics, metagenomics, epigenomics, and methylomics. So genomics is for a particular cell, let's say Clostridium thermocelum strain 1, 2, 3, something like that. When I sequence each and every ORF, each and every nucleotide in its entire genetic material, it's called genomics. When I take an environmental microbial community and I sequence all the genes, all the ORFs, all the nucleotides present in that environmental microbial community, it's called metagenomics. So metagenomics would be, for example, all the bacteria are sequenced, all the archaea are sequenced. Now epigenomics, when all the epigenomes are sequenced. Methylomics, when I am looking at all methylated DNA. Now when it comes to RNA sequencing, I can also do RNA sequencing, and RNA sequencing is very, very useful. Think of it this way: DNA is the potential to make a protein, but DNA does not necessarily say that protein is being made, because it's possible that the gene that makes a particular protein is down regulated and not allowed to express, or it is damaged and it will never express, or until it gets its signal it will not be expressed. So just because the DNA is present doesn't mean that cell carries out the activity. For example, a particular cell might have the gene required for degrading beta lactamase by antibiotics, but it's not expressing them, so it won't have resistance to beta lactamase based antibiotics through that mechanism, degrading them, right? So if you look at RNA, we know that which genes are being upregulated, which genes are actively being transcribed into mRNA. So that's why RNA sequencing is very important for us, and when we sequence all RNA present in the microbe, it's called transcriptomics. So transcriptomics is more informative than DNA analyses like genomics or metagenomics, but there is a big challenge here. In transcriptomics, one of the biggest challenges is that most RNA are short-lived, and then RNA vary in their longevity too. Some RNA will destroy faster, some mRNA will live longer. So as time proceeds, the delay in our sequencing proceeds between sampling and actually sequencing it, we'll have a very different picture maybe than what was actually present when we sampled it. So there are some limitations with transcriptomics, and there are some ways out. We add some reagents to prolong the life of RNA. We store RNA at -80 to prevent its degradation. But if successfully done, transcriptomics is very promising. Now let's look at protein. Protein, we look at proteomics, translatomics and interactomics. Now proteomics is I look at all the proteins in the system and I sequence all the amino acids. This is proteomics. If I do proteomics analysis for all microbes present in an environment, it's called metaproteomics. And this is very tricky because it is very hard to isolate protein, purify them, and then do metaproteomics on them or proteomics on them. Translatomics is when I look at all the proteins that are freshly translated. So what is being translated right now? What activities are being done right now? Interactomics is when I look at the interactions between the proteins, RNA and DNA. So interactomics actually includes the DNA analysis, the RNA analysis and the protein analysis, and this is very important when I am interested in regulation, because some of the regulation is carried by enzymes. So there are some enzyme inhibitors that don't allow other enzymes, other proteins to do their job. Sometimes it's RNA molecules, sometimes it is other molecules that actually upregulate or downregulate proteins, genes. So interactomics looks at the interaction between all three. Then we have metabolites. So metabolites would be, for example, acetate, glucose. So when I look at these metabolites, so they are the breakdown products or products of anabolism or catabolism, and I characterize them completely. It's called metabolomics. Now metabolomics is very, very important for environmental engineers and biotechnologists. Think about it this way: I want to make butyric acid out of cow dung. So I definitely need to do metabolomics to understand usually what are the different degradation products of cow dung, what microbes are involved in it, what are the pathways, that chemical pathways they are going through, and then I can try to tweak them and to make sure that they make what I want them to make. So this is why metabolomics is very important. And then we have glycomics, when we are only looking at the glucose. So we are characterizing glucose in a microbe, when it is making, how much it's making, etc. at any given time. That is glycomics. Now when I am looking at organisms from a higher picture, I have microbiome, virome and mycobiome. So microbiome is when I am characterizing not just when I am characterizing microbes from different domains. So 3 domains: prokaryotes, eukaryotes and archaea. So when I'm characterizing all of them, when I'm characterizing all the bacteria, all the archaea and all the eukaryotes, including higher order of life like fishes and algae, then it's called microbiome. So for example, I often talk about drinking water microbiome in my classes. So what I'm referring to is all the organisms present in drinking water. They could be protozoa, they could be virus, they could be bacteria, they could be algae, they could be anything. Virome is when I am looking at all the viral particles present in my sample, and microbiome is when I am looking at all the fungal particles present in my sample. Alrighty dear friends, now we have a wonderful method of getting an idea of expression. So I am interested in which gene is getting expressed or not. So this is microarray preparation, and I will very briefly go through this. So in microarray preparation we do not need to sequence, but what we already know is we have certain genes of interest. When these genes are transcribed, they make RNA, and their RNA, how they look like, we know. So we take a chip like this, or this is a PA surface. We use photolithography to etch them, activate them. We attach these small primers to them, and we allow synthesis of the RNA that are of interest. And when I attach, so I can in the same singular plate, what I can do is I can attach multiple primers, so I can have multiple templates. Now when I flow my sample through it, the complementary strain would attach with these RNA strains, and I will know what is being transcribed, what is being expressed, what is being translated by getting a signal. And there are many ways of doing it, how the hybrid microarray system is prepared, but basically it works on DNA hybridization. So for example, let's say this is the DNA that I'm interested in, and I know its perfect sequence, so I can have a single stranded DNA of the right sequence, and this is the other environmental DNA that I've isolated, and I want to know if it is this DNA of my interest. So what I can do is I can allow them to interact, and if they are perfect complement to each other, they will hybridize. So they will form hydrogen bonds, and they will form one double stranded DNA. And when this is formed, if my single stranded was fluorescent probe, when they form bonded double stranded DNA, the fluorescent probe will be released and the fluorescence will be measured. So this is how microarray system works. Now in my microarray, what I can do is I can make these genes of interest, multiple genes of interest, and I can attach them, ligate them to my surface, and as I flow my sample, whatever has the perfect complement will attach, and the corresponding fluorescent signal will be released. So then I can read my chip and I can say, alrighty, here we saw a signal, here we saw a signal. So in this sample, these expressions were happening, these RNA molecules were present. So on the right panel you have treated cells, on the left and here you have control cells. So DNA or RNA is isolated from both samples, it's transformed and amplified into fluorescently labeled cDNA. So if it is RNA, it will be converted into fluorescently labeled cDNA. If it is DNA, just fluorescently labeled DNA. And then they have labeled fluorophores, as the name suggests. Then they are from the control, and this is hybridization, by the way, in the right panel. The control and the treated cells are mixed with each other. When they hybridize, they are hybridized on microarray plates, and they are scanned. Wherever I get a signal, I know hybridization happened, so the match was found. Now this is a very beautiful technology. It's a gene chip technology, and I think it's very important to understand this. Where we are headed now is to a highly personalized medical system. We know certain diseases such as cancer, for example, does not manifest similarly in two people. The immune system of two different cancer patients will behave very differently when it's trying to body is trying to feed the cancer. That's how cancer works. And when we add cancer drugs to kill the cancer cells, the body finds ways to protect the cancer cells, because it treats cancer cells as its own very important cells. So when this happens, the genetic sequences of the cancer cells mutates and undergoes different changes, and these changes determine the resistance of cancer cells to any anti-cancer drug. So in gene chip, what we can do is we'll have the genome of the entire human put here. So I have the genome of entire human put here, and then I can read the genome, and I know what is going on in the human cells, what's going on in the cancer cell, what RNA is being translated, what gene is being transcribed, expressed. So in this gene chip, I'll have thousands and thousands of complementary RNA molecules attached, so very small. And when I flow the sample over it, so this is gene chip, when I flow the sample over it, a human sample, blood sample, stool sample, fluid sample, whatever, and then the hybridization would happen, the perfect complement would hybridize and release a fluorescent signal like this. So this is a blown up image of very small portion of this. So wherever I get a signal, the computer can read it and say, okay, these RNAs are being expressed. So if I know some fishy activity is happening here, I'll know exactly what the resistance mechanism is there of this particular cancer patient. So let's look at this particular example from the McGraw-Hill Company book. This is their two leukemia patients, leukemia patient 1 and leukemia patient 2. I take their bone marrow cells, I take the DNA out of it, and I put it on my microarray, which have these thousands of thousands of oligonucleotides that might be complementary to this DNA, and wherever the hybridization happens, the computer checks. Similarly, here in leukemia patient 2, I can again take his samples, his fluid bone marrow cells, put them on the same chip, and notice what his signals look like. So this is the SNP profile. Now, as the leukemia is progressing, there are mutations happening in the genes. So what I'm getting are mutation usually start with single nucleotide. Well, there are many ways of mutation. One of the ones that is presented here is single nucleotide polymorphism, where a single nucleotide changes. So wherever there is a single nucleotide change, there you have a green signal, here you have a red signal. So what you can do is you can find out the similarities. Okay, in both leukemia 1 and leukemia 2 patient, these yellow ones are similar and the green and red ones are unique. So here you can get an idea of how leukemia progresses overall. If you do with enough patients, you can also get an idea of how leukemia expresses itself very differently for each of the patient. So my dear students, I will end it here, and in the next lecture we'll go ahead and we'll talk more about environmental genomics. That's all for today. Thank you.